Hello, everyone. Welcome to the second day of the Canadian Computational Neuroscience Spotlight. We are going to have session 3 for trainees talk, and Carter is going to start as the first trainees talk. Please, Carter, go ahead.
Thank you. Yeah, a big thanks to the organizers for having me and for all the presenters for making this great conference so far. I'm Dr. Carter Johnson. I'm a postdoc in math at the University of Utah. I'm in the computational neuroscience group led by Professor Ala Borsia. And today I'll be talking about how astrocyte calcium activity can affect the excitability of nearby neurons. So as we saw in Mauricio de Pita's tutorial and Evan's talk just now, astrocytes can have effects on nearby neurons, right? They're specialized glial cells, they're throughout the CNS, and one of their major roles is to wrap synapses to form tripartite synapses, right? So the astrocyte will go up to a neuron-to-neuron connection, wrap it partially, and that gives it direct access to the ions and neurotransmitters in the synaptic cleft, okay? And so what we're interested in is, how does an astrocyte affect the ionic concentration in that synaptic cleft, right? And then in that effect, how does that change the excitability of nearby neurons, okay? So to do this, we look at a detailed model of astrocyte dynamics. So we model the tripartite synapse, where we're looking at the extracellular space here in the synaptic cleft and all the ion concentrations and neurotransmitter concentrations within there. And then the astrocyte with all of its various channels, transporters, and leaks that interact with that synaptic cleft, okay? And so there's a few pathways here through which the astrocyte can modulate neural dynamics. The first is that it can uptake potassium ions through these inward rectifying potassium channels, KIR41. Okay, so this is a channel that preferentially will remove elevated potassium from the synaptic cleft, right? So the elevated potassium could lead to enhanced excitability for the postsynaptic neuron. And so that one of the astrocyte's jobs is to clear that elevated potassium and return things to baseline levels. Another pathway is uptaking glutamate through the glutamate transporter EAT2, or sometimes it's written as GLT. And glutamate is an excitatory neurotransmitter. It's released by the presynaptic neuron and attaches to receptors on the postsynaptic neuron, which cause that postsynaptic neuron to fire an action potential. But glutamate is not taken up by the postsynaptic neuron. It actually unbinds and could rebind after some recharge time, unless it diffuses out of the cleft or is taken up by the astrocyte. So one of the other astrocyte roles is to clear that glutamate so that we don't have runaway excitability in the synaptic cleft. Another pathway that it could affect the ion concentrations is that elevated astrocyte calcium levels through this IP3 and endoplasmic reticulum pathway can drive sodium-calcium exchangers, right? So if calcium is elevated, you can exchange that for sodium, which can then drive a sodium-potassium pump to exchange that sodium for potassium to help clear that elevated potassium again in another pathway. And that intracellular calcium is increased via this IP3 signaling pathway. And so we're building on previous work from our group here. So Handy and Terry a few years back made this nice set of experiments and model that looked at how different IP3 signals can cause different shaped calcium transients and calcium responses inside that astrocyte. And so the question we're looking at here is then, okay, how do those different calcium responses lead to an interaction with the ions in that synaptic cleft and then the excitability of those nearby neurons? Okay, so to do that, we need to look at adding on these new model components where we look at various transporters, exchangers, and pumps that will relate that calcium increase to changes in the other ionic species within the synaptic cleft. So lots of other papers, models have looked at, kind of this picture in various ways. So some models have looked at sodium and potassium together. Some of them have just looked at glutamate. Some have looked at glutamate and potassium and sodium. But to my knowledge, I don't think any have looked at calcium, sodium, potassium, and glutamate kind of all in one model. So that's what we're trying to do here. So the bulk of my work over the past year has been looking through what's been done before and trying to pick and pull out different components from different models that capture the right behavior. So we're looking for each of these various channels and transporters that we're adding to the model. We're re-examining, what those expressions for those channels or transporters look like in previous models, and refitting them or tweaking the expressions so that they fit the astrocyte-specific data that we have from many of these experiments here, and also just fitting well together in this kind of full model. Okay, and there's lots of challenges involved in this process that I could talk about for a long time, but I only have 8 minutes, so I'd like to get to some exciting preliminary results that we've made from this new model. So what we've seen in just adding these model components is that astrocyte calcium activity can increase that extracellular potassium clearance rate. So when you have elevated potassium in the synaptic cleft, maybe from repetitive firing or maybe some more pathological seizure conditions, the potassium is elevated and it needs to be cleared by that astrocyte and returned to the resting potassium levels. So that's cleared through the inward rectifying potassium channel and the sodium potassium pump. Okay. Adding in this extra calcium response and all these other channels actually ends up speeding up that process, right? So with this calcium response, this potassium is cleared much faster. There's a bit of an overshoot, and it's cleared faster compared to this elevated potassium with just the standard astrocyte clearance processes. And so what's happening here is that increased internal calcium through the IP3 signaling pathway, but in this example, we've just kind of prescribed a calcium forcing function. But what that leads to is an increase in internal sodium through the sodium calcium exchanger. So that increase in calcium leads to an increase in sodium because they're just exchanged. And then that leads to an increased external potassium clearance through the sodium potassium pump. So we have more internal sodium, so now we can pump in more external potassium. And that helps clear this faster. And I talked about a little bit already that this elevated extracellular potassium in the synaptic cleft can increase the excitability of nearby neurons. So what we want to do here is say, okay, looking at this astrocyte, we have these two kind of different effects on the nearby extracellular potassium, right? When I have the calcium response, it clears faster. When I don't have the calcium response, it clears at a slower rate. And that if I look at these potassium transients at the same time, I can compare the excitability of a nearby neuron, while it's clearing this elevated potassium. So astrocyte calcium activity is going to change the excitability of nearby neurons here. So that change in extracellular potassium is going to lead to a change in the potassium reversal potential of that nearby neuron. So here it's basically the same plot, but now I'm plotting the reversal potential for potassium for a nearby standard Morris-Lacar neuron. And we can see kind of the same story, right? The elevated potassium with the calcium response has a much faster decay to baseline resting reversal potential levels. That's going to change the excitability of a nearby neuron, right? So here I have two standard Morris-Lacar neurons. One of them is exposed to resting potassium levels. The other is exposed to this elevated potassium without the calcium response at that particular time in the transient. And if I apply the same short step current, the neuron that's exposed to elevated potassium is going to spike, and the one that's exposed to resting potassium levels is not going to spike. And that faster clearance of potassium with the calcium response is going to return this excitability back to baseline levels. Okay. So the take-home message here, the astrocyte calcium activity can change the excitability of nearby neurons in subtle ways, and that can have possible consequences for network level dynamics. Thank you.
Thank you so much, Carter. We have one question. Can you see the question in the, please go ahead. I mean, can you read it first? And then
does it mean that an astrocyte works as a local garbage man? Yes, I guess that's how I describe it. Yeah, it's responsible for clearing glutamate, clearing potassium, returning things to baseline. Yeah, definitely.
Perfect. Thank you so much, Carter, for your interesting talk. I would say it's not easy to deal with the astrocyte, and I hope that one day we have very kind of abstract models that we can simply integrate to the other neuron models, and then that day is not that far. Okay, thank you again, Carter. I'm going to invite Julio, our next speaker. So let me find Julio. Yes.
Hello, can you hear me?
Hi, Julio. Yeah, it's everything's fine. Please
Cool.
share your screen, and then I'm going to focus on your screen.
Great.
It's perfect. Yeah, please go ahead.
Can you see my screen? Yep.
Yes, everything's fine. Go ahead.
Okay. So, hello everyone. My name is Giulio Bonifazzi, and I'm a PhD student at the Bus Center for Applied Mathematics, where I work in the group of Maurizio De Pitam. Today, I will show you our results about how the alteration of astrocytic glutamate transporters drives tractability at the onset of Alzheimer's disease. Alzheimer's can be very well represented in this series of self-portraits where this painter on the left has a completely healthy brain, but then gradually and step by step developed the disease, up to the end on the very right, where his dementia is already at a very advanced stage and is not even able to, let's say, recognize and depict himself. But unfortunately, even if let's say, in this case, we have this gradual representation of Alzheimer's, this is not the case when we look for biomarkers of Alzheimer's disease. In fact, among the known biomarkers that we have, for example, there is the amyloid beta accumulation, where amyloid beta is a molecule that in the healthy condition of our tissue, of our brain, so let's say the left of this plot here, has a very low concentration. But then we know that when dementia is already at a very advanced stage, this concentration can be very large, and so even form some solid plaques in the extracellular space. But the problem is that we don't know how we actually progress from one stage to the other. And so here comes our research question that motivates our work. That is, how do we switch state? How can we actually map Alzheimer's from one side of the left of this plot to the other side of the right? In order to look at this question, we move to a synaptic level. And in particular, we look at the interaction between amyloid beta and calcium dynamics. So here, what we know is that, first of all, amyloid beta can modulate, can increase the calcium influx into neurons. But then this calcium concentration can as well increase and promote amyloid beta production. And so you understand that we have this positive interaction, so a positive feedback loop between amyloid beta and calcium. This positive interaction has been studied in, let's say, in a dynamical framework in this work by de Calouvet-Dupont back in 2013, where with using this dynamical framework, they saw that this positive feedback loop translates into a bistable system. What do I mean by bistable system? So if we look at the bifurcation plots here on the right for the amyloid beta concentration and the calcium concentration, we see that for in a certain range of value of amyloid beta production rate, we have two stable branches that are these two continuous lines, one on the bottom for the healthy condition and one on the top for the pathological condition. And what does it mean to have bistability here? It means that even if, for example, let's assume we start from the higher branch, we can still push back our system into an healthy condition. And so you understand let's say, the translational relevance to have bistability in this system. But this is only half of the picture. In fact, we need to consider a synaptic level also neurotransmitter release and how this neurotransmitter release interacts with the firing activity. In fact, when glutamate is released from a presynaptic terminal, it binds the postsynaptic receptors so that the activity and the signal can keep going on. But the more we stimulate this firing activity, the more we will also release. And so again, another positive feedback loop, also in this case, but between glutamate and firing activity. But this time is actually different. In fact, we have a third actor in the game, which is the astrocyte, that in this case that surrounds the two terminals and sequester the molecules of glutamate from the synaptic cleft through these glutamate transporters of type GLT1. And from experiments, we know that these transporters the level of these transporters is modulated by the amyloid beta concentration. And you can understand that if amyloid beta reaches values, let's say, too large, can decrease these transporters to below a certain threshold. And so this may result in a pathological condition because of too much glutamate in the synaptic cleft. This pathological condition is usually also referred as excitotoxicity. And this is exactly the case when we look and we study again, we translate this biological mechanism into a dynamical framework. And so here on the right, again, you see the bifurcation plots, but in this case for extracellular glutamate and the firing rate. So in this case, you see that when we have a small value, a small level of transporters, we only have this upper stable branch for the pathological condition. So with large glutamate concentration or large firing rate. As soon as we increase this level, again, we have this bistable condition, which means there's this coexistence of both the pathological and the healthy state of our system. Now, when having these two bistable systems separate, we can actually combine them. In particular, we combine them by describing, by integrating the calcium modulation into the glutamate release, as calcium is promoting the release of vesicles from the presynaptic terminal, and also how the firing activity is also modulating the calcium influx in neurons. And when we when we put together, when we combine these two subsystems, we observe something even more interesting than before. In fact, we observe that the two bistability translate into a tristable system. This means that instead of having only two stable branches, we have a third intermediate stable branch in the middle. But depending on the parameter the parameter set that we use, we can result in different conditions. For example, in this case, you observe that this intermediate stable branch is for moderate values of extracellular glutamate. Whereas for amyloid beta here on the right, this intermediate stable branch has very large concentration. So this means that in the progression of the disease, we are developing first amyloid beta accumulation, and then excitotoxicity or glutamate concentration is kicking in. But if we use another set of parameters, we can actually get the opposite condition, which is before we get excitotoxicity, as this intermediate state of the disease has a very large concentration of glutamate, and then we develop amyloid beta accumulation, as this intermediate state branch has pretty low amyloid beta concentration. But besides which of these two pathways, let's say, of the disease we follow, the main message here is that we have this tristable system, which means an additional, additional threshold, an additional point to attack the disease with some therapeutic intervention. And with this, I conclude my talk. And I just still another 10 seconds to tell you that starting from September, I will be in Toronto and I will join as a graduate student, the physiology department. So as many of you are in Canada, I will be happy to meet you.
Thanks. You, Julia. It's great. And we're so happy to have you here. So there is one question. If you go through the ask questions section and you read the question and please answer it.
Oh, sorry. At what time scale does amyloid beta change the expression of GLT1? Okay, so this is a complex question. From experiments, we know that amyloid beta is changing the expression of GLT1 at two different scales, either minutes, so a short application can already change the level of transporters, but we also know that in terms of hours, we may have a change in the transporter expression.
Great. Thank you so much, Julio. Thanks again for your
Thanks.
talk. And yeah, I'm going to invite our last speaker for the trainees talk session. Let's go. Hey, Pascal.
Hello.
How are you?
Fine, and you?
Good, good, thanks. So your first name is Pascal?
Do you hear me well?
Yeah. Go ahead and share your screen.
Okay, share.
It should be on top of your photo. That's perfect. Good. Please go ahead. We can see everything. Can you? Yes.
Do you see it?
It's coming.
Do you
Not
still
yet.
see it?
No, it's all black, but we could see the before you did the last
Okay,
change.
before I can stay.
Yeah.
Yeah, like that. Maybe it's.
Yeah, it's not full screen, but I think it is very visible.
It's okay.
Yeah. Please go ahead.
Okay, so hello and thank you everybody, and special thanks to the organizers for allowing trainee talks. So I'm Pascalson, and I just finished my PhD, and I will talk about a joint work with Etienne Tarin and Romain Bels. And I will talk about a new stochastic STDP rule in a neural network model. So first, I will give a brief overview of my talk. So what we were interested in this work was the interplay between the synaptic and neural dynamics. And in particular, we wanted to study it with mathematical tools. So this is really a hard task, a difficult task. So that's why we proposed a new stochastic STDP model, which were more tractable and but has still interesting biological features. So then we study it using a slow-fast analysis based on the difference of time scale between the neurons and the synapses. And finally, we study the long-time behavior of the synaptic weights dynamics that we obtain with the slow-fast analysis. So first, I will give some detail on the spike timing dependent plasticity mechanism. I think a lot of you know it, but I will go it quickly. So I will go quickly. So basically, when a neuron I, the spikes of the neuron I will cause the spike of a neuron J repeatedly, the weight between the neuron I and the neuron J will have a tendency to increase. That's what we call LTP for long-term potentiation. And then in the other case, we have a possible decrease of this weight between the neurons, which is called LTD for long-term depression. So this weight changes depends on the time delays between the spike of the neurons, and it has been shown that it's follow like exponential curves depending on this delay. And another thing on this experiment of BNP is the fact that we need a large number of spike pairs before we observe a change in the weights. So we can model this phenomenon by two methods. First, we can change the weight at each spike pair, but with a small increment, which will lead to a macroscopic change after a large number of spike pairs. or what the method that we used is the other one. So at each spike pair, we can make the weight will have the possibility, sorry, to make a macroscopic jump, but with a small probability. Then we obtain stochastic plasticity and discrete synaptic weights, which is more biologically plausible than continuous weights. So I will present the model, which is the stochastic Wilson-Cohen model in which we implemented STDP. So it's a model with n interacting neurons described by their potential, which are binary, and the synaptic weight matrix, which we will suppose will take integer values. So first, I will give the dynamic of the weights. It will depend on the time since the last spike of the neurons, and so at each time the neuron I spike, the weight, all the weights, so the incoming weights in the neuron I will have a probability to increase depending on the Sj and the current weight value. And in the other way, the outgoing weight, Wji, will have a probability to decrease. And this function p plus and p minus will depend, will have an exponential form, such as in the B and Poo experiment. So now we see that we need the variable st, which is the time since the last spike of the neurons. And this variable will be described like this. When the neuron spikes, so I will say that we will say that the neuron spike when the v, its potential jumps from 0 to 1. So we'll see that when there is such a jump, the corresponding s will come back to 0 and then increase linearly. So how does this v evolve? So it's simply, it will jump from 0 to 1 with a function in function of the current weight matrix and the current neural states, and it will come back to from 1 to 0 with a constant rate beta. So with this model, now we want to use the fact that the plasticity is really slow compared to the neural dynamics, and we will introduce for that a parameter epsilon, which is small, in front of the in factor of the probability, the weight, the jumping rate probability. So now we look at what's happening when epsilon tends to 0. So first, when epsilon tends to 0, the weight will not change. So we are interested in the behavior of the process when W is fixed. And we show that Vs will converge exponentially fast to a probability distribution that we will call PW. Then we will look at the dynamic of the weight on the accelerated time scale, T over epsilon. And we show that on this time scale, the weights will converge to a process that we can describe thanks to the invariant measure PW. And here, so the EIJ are is the canonical basis of the n by n matrix. So now we have this limit process. We studied the long time behavior of this process. And in particular, we found condition for which we have a convergence of our limit process W bar, and so convergence to its unique invariant measure. So I will show you how to use this condition in a real network. So we have to compute the R plus and R minus. And here we see that this, with the exponential function for the P plus and P minus, the R plus and R minus depend on the Laplace transform of PW. So in the case of two neurons, we are able to compute this Laplace transform, and we can find some parameters for which we know that the weights should converge, so as in the left, or diverge as in the right. So the parameters are found with the limit process, and this is an illustration for the initial process with a fixed epsilon equals to 0.01. Then I will just show you that when we increase the number of neurons, this condition of convergence is still working, and we have even less restrictive conditions on the parameters than for two neurons. So here we see that we have until a plus equal 0.6, we have convergence of the weights, and after, so between 0.5 and 0.6, we'll have the divergence of the weights. So I will finish on this slide, and we wonder if we can prove this fact that when we increase the number of neurons, we can still study the behavior of the weights with only two neurons. So I will just finish with perspectives. So we would like to extend the model and perform a mathematical analysis on this extension. And we would like also to investigate the memory capacity of this model, and finally pursue the mathematical analysis. Also, this mathematical analysis can be done also combining the mean field and slow fast analysis. So we did a mean field analysis also on this model.
Thank you. Thanks a lot, Pascal, for your talk. Is there any question from the attendees? There is no question actually in the section for asking question, and I don't see any other question in the chat. So yeah, that's it. Thank you so much, Pascal, for your interesting research talk. Yes, and I'm going to, so don't forget to join us at 10 for the tutorial talk by Dr. Carmen Canavier. And yeah, so see you guys at 10.
See you, thank you.